Hi learners, I am Dr. Gurunathan, Christu Jayanti College, and I welcome you all for today's session which focuses on genome sequence database. When you speak about the database, this database is operated by the National Center for Genome Resources. In short, it is called as NCGR, and basically this is a relational database which is publicly available, and it contains nucleotide sequences and associated biological and bibliographical annotation. And we should see that this database, it contains both genomic as well as expressed nucleotide sequences from essentially all organisms from which some sequence data has been determined. And when you speak about the expressed nucleotide sequences, we are speaking about the expressed sequence tags, ESTs, associated with different organisms. And when you speak about this database, it requires a few steps. The first step is the data acquisition. So approximately 5 million sequences of the databases are submitted to the public domain, and you also see that it is having information associated with EMBL and GenBank, and these are all the main portals where the sequence submission and analysis can be done. And moving down to the data access, which is to be continued, as far as NCGR is concerned, it has a variety of tools to identify and retrieve sequence of our interest. And you can access this NCGR, the website called www.ncgr.org. And you see that you can search this database in terms of organism name, in terms of gene name, or in terms of other keywords that are associated with the search term. And you can also employ Boolean operating system, and because it is defaultly incorporated in the tool, it is very easy for you to search the database. Similarly, you can see that there is a tool called excerpt tool which allows the users to find out their sequence of interest. And there is also another tool which is a customizable tool which is called as sequence viewer, which allows researchers to navigate and examine the sequence present in this database. And we can also visualize these sequences with their biological features. And you can see that all these tools are accessible from ncgr.org website. Moving down to the other aspects related to this NCGR, you can see that it has a lot of information pertaining to the legume plants, which is called as legume information system. You also have something called as genome context viewer. Similarly, you have something called as legume federation. You also have the Arabidopsis information resource. In short, it is called as TAIR, and this was formerly developed and hosted by NCGR, whereas now it is being operated by another organization called Phoenix Bioinformatics. It contains all the necessary information related to Arabidopsis, the model organism. And you also have Alpheus, you also have UMI analysis, you also have transcriptome assembly pipeline BPA 2.1.0. Similarly, you also have integrated system which is called as ISIS. You will also have comparative map and trait viewer CMTV, which is also commonly employed when you are comparing different organism genome and when you want to understand which trait is exclusively present in which organism. And you also have what is called as XGI. This is a genome initiative for species X analysis, and it's a pipeline or a web interface that is also existing. So you see that the entire NCGR has so many tools and so many databases and so many resources associated with it. And when I think about NCGR, I should definitely look into three different aspects of the NCGR content. One is the genomes of the organism, the other one is about the transcriptomes of the organism, the third one is about the metagenomes of the organism. You can study genome of organism. For example, I want to study about the epigenetics and the chip sequence analysis. Then the information pertaining to those experiments are available in this genome. Or I want to perform genome assembly and scaffolding. That is also possible. You have tools and you have databases which have the necessary information in them. Similarly, I want to use the genomic information for drug discovery. definitely we can do it. Or if you want to undergo, or if you want to study more detailed information about the genome itself, where you're comparing different genomes and trying to understand what features are common to them, what features are different to them, definitely that can also be done using NCGR. And when you move down to the transcriptomes, there also we can employ different tools present in this genome resource, and you can perform so many analysis that it can include single cell transcriptomics. If you want to study about the RNA-seq expression, pathway and enrichment analysis, that can be done. Similarly, if you want to do for small RNA analysis, that can also be done. And when we move down to the metagenomes to be studied using NCGR, you can perform either a whole metagenome sequence analysis, or you can specifically focus on the 16S or the 18S microbial community analysis. And as we all know, sequence analysis requires a very important tool called as BLAST. Apart from BLAST, there are also several other tools. For example, you have an algorithm called Smith-Waterman algorithm and a frame search, which is also available in public. These are also useful for us to understand the sequence and their interesting information. And you also have specific prediction tools associated with this database, which helps us to study it better. Particularly, this is having improvements to tools like Grail and Gene Finder, which are very commonly employed to understand the gene prediction present in the given DNA sequence. Similarly, you also have something called as MarFinder, which is a very popular tool which helps us to identify regions that often mark concentrated areas of transcription factor binding. So like this, you have plenty of tools associated with this database, and we can definitely explore them as per our needs. I end up my session with this discussion. I hope you had an interesting session today. We will meet again in another interesting session later on. Take care. Bye.